The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everybody, and welcome to the 24th Annual Green Chemistry and Engineering Conference and to our third and final virtual webcast. We're delighted that you could all be here, and we're very much looking forward to having you join us for next week's conference, beginning June 15th and going through the week. These kinds of events, conferences, cannot happen without the kind generosity of our sponsors, and I'd certainly like to thank all of our sponsors for sponsoring the conference. And I'd also like to thank the Pharmaceutical Roundtable, who have been a consistent supporter, very major supporter of the ACS Green Chemistry Institute, as well as this conference. There's lots of sessions through the week that they have organized and will be participating in. This is really a unique collaboration. You'll hear a lot about it and throughout the day today, but we're just very thankful for that collaboration we've had. Next slide, Jenny. I would remind everybody to please ask questions during each speaker's presentation so that we can get those ready to be asked at the end of the speaker's presentation. And with that, I'll turn it over to Juan Kohlberg, who is the organizer for this session.
So good morning and thank you, David. Welcome all to today's symposium on the American Chemical Society Green Chemistry Institute Pharmaceutical Roundtable, past, present, and future. We really appreciate you all joining us under these extraordinary circumstances. Hopefully you all keeping yourself safe. And we'd like to especially thank all our speakers for staying with us through all the different changes that we had along of organizing this symposium. We're very excited to share with you all a series of great presentations. And I think that you will agree with me that we have an amazing lineup of speakers representing of what the Pharma Roundtable has been doing for the last 15 years. The Pharma Roundtable started back in 2005 as a partnership between the Green Chemistry Institute and a group of pharma companies. And now today in 2020, the Pharma Roundtable have grown to up to over 30 members up to 2020. Recognizing that the research, important on the research for advancing the green chemistry agenda, and to having the opportunity to reach out to other part of the chemical industry, we have included members that represent what our external network is today. So this part of our supply chain has been included, as well as members, as well as other industry where chemistry is key in our business. And in this slide, we can see how we have grown in the last 15 years with representation, as I mentioned, from all the pharma industry together with other part of our supply chain and external network. Our membership have grown to such a reach that have now a network in Asia, Europe, and the US. And we continue working with government and academia in these areas to address what we need are the solutions to develop greener processes in our operations. Part of that, what we have been doing during this year includes several programs where we have supported and sponsored technical and key technologies aiming to develop the greener processes and bridging the gap between academics and industry. Our grant program have funded over $2.4 million of research including 28 research programs, and have resulted on the leverage of 1.3 million from other government agency. Today also, we're lucky to have one of our former grant recipient, Professor Jeff Byers, which also will be a speaker on this session. And later you will see some of the great work that he is doing with his group. As part of the collaboration that we have been having again across pharma, academia, and government agency, we have done a lot of work around how to facilitate our scientists to make better decisions, aiming to develop greener processes all the way from discovery to commercialization of manufacturing activities. During next week conference, as David mentioned, you have a chance to attend several presentations from the Pharma Roundtable member, where they will highlight in more detail how we have accomplished developing of some of these tools and how are they being applied in our industry to improve our environmental and sustainability performance. Also today, later in this session, we will have the presentation of the 2020 Peter J. Dunn Award for Green Chemistry and Engineering, Impact in the Pharmaceutical Industry. This award was named for a former Roundtable co-chair, a Pfizer colleague, and a good friend, and more important, a great advocate of the green chemistry in industry. Also later, at the end of the session, we will have the pleasure to have Dr. Frank Gupton, the 2019 award winner, speaking for us today. But certainly, we're here really to listen to the great lineup of speakers that we have, and I don't want to keep you waiting for them. So this agenda reflects what we're going to be doing today, starting with a presentation from Dr. Mike Kopak from Eli Lilly, followed by the presentation of the Pete Dunn 2020 Pete Dunn Award. And then we'll have Dan Bailey from Takeda, following with Megan Shand going before our intermission. After the intermission, we will complete the presentation with two fantastic presentations from our academic representatives, Professor Jeff Bayer from Boston College, and as I mentioned, Dr. Frank Gupton from the Virginia Commonwealth University, with some closing remarks at the end of the session. Again, I really want and hope that you will enjoy this conference, and without any further delay, I'd like to introduce our first speaker. Today's first speaker is Dr. Michael Kopak, who is a Senior Research Advisor for Eli Lilly and Company. Mike earned his PhD in organic chemistry from the University of Virginia back in 1995 under the guidance of Professor W. Harman. He completed his postdoctoral studies in natural product synthesis at the Colorado State University with Professor Al Myers. Mike started his professional career back in 1997 with Roche, and then worked in different processes, including the nelfinavir, Senecal, and furbirazepide first synthesis, moving later in 2001 to Eli Lilly, where he has led several small molecule API projects, including Aluminet, an important rheumatoid arthritis that currently is being evaluated for COVID-19 treatment. Mike also recently established a synthetic peptide division at Lilly, and is leading the effort to advance sustainability practices in this important discipline, which is a critical unmet need. Throughout his career, Mike has led research team applying green chemistry and other greener technologies, and has represented Lilly in the Pharma Roundtable for many years, where he have actually served for 2 terms. It's the only co-chair that has served 2 terms, and the rumor is that he is looking for the 3rd term pretty soon. After that, certainly Mike has been a key partner with the Pharma Roundtable to grow our membership, over 30 members right now, and he will be presenting in the presentation around the early days of the American Chemical Society Pharmaceutical Roundtable, establishing a pre-competitive consortia, which is thriving 15 years later. And with that, I have a pleasure to pass it to Mike.
Thanks, Juan, for that really great introduction. And yeah, related to your question about looking for another chairmanship with the roundtable, I think I might pursue that when the roundtable hits its 30th anniversary. So that'd be a good objective, I think. So this is one of the presentations that, the presentation I was looking most forward to all year, because this is such an important research group, the Pharmaceutical Roundtable, important industry group. And I think to lead this off, just want to give a little bit of background on green chemistry. And again, probably 99% of the people in the audience don't need this, but for the 1% there that is here today, that is seeing green chemistry for the first time, just want to provide a little background there. What is it? Green chemistry, certainly. There are a lot of definitions described on this page. You hear synonyms, sustainable science, and there's a lot of other words that can be used to describe green chemistry, like shown in the bottom left, and they all can be correct. Catalytic, avoid waste, sustainable, safe, non-toxic. But one of the ones I really like the best is from Nobel laureate Nuriori, where describes green chemistry is not just a catchphrase, but just an indispensable principle that guides chemical research and really is a foundation or a pillar for civilized society to advance. So a really, really holistic philosophy it encompasses as well. And my very, very favorite is the box shown here on the bottom right. Green chemistry is simply the best chemistry. It's the chemistry that you want to do that is going to be environmentally sustainable. And so what group do we have that supports green chemistry? We have the Pharmaceutical Roundtable, and Juan and David actually showed this in the intro slides, where we're up to 37 companies if you conclude the affiliate and associate companies that are associated with the Pharmaceutical Roundtable. And this is just a really, really important slide, because there's really no other scientific group like this out there that is working together collectively, all the oars steering in the same direction, to catalyze green chemistry and engineering in the pharma industry. And this has been going on since 2005. And I'll move to the next slide here. So the ACS Pharmaceutical Roundtable essentially has been involved, has built itself up right now to where there's so many different activities this group has been involved with, is involved with spreading the message of green chemistry and engineering. And this just highlights some of the areas that we are in fact involved with. So if you look on the top left here, one of the things this group does is develops tools for innovation in chemistry, including solvent guides, reagent guides. And these are tools that aren't just available for the pharma industry, but they're publicly available. We post these on our website, and any scientist or anyone interested can actually access these. And I'll show an example of this coming up in a minute. But you see here, we also have a big focus on the right of advancing research, getting our message out about what we are about, and then funding external research. It's a big part of our mission. Our group does, in fact, collaborate globally. We've had many efforts in recent years where we've expanded our outreach to India, to China, the Pharmaceutical Supply Chain Initiative, and we collaborate with other external consortia as well. So really, really big mission by this group, and really on any one of these individual topics here, you can have a full presentation of all the work that is going on. So I just want to highlight one innovation tool. And again, this is actually publicly available to anyone, and that is our reagent guides. And if you see here, we currently have 18 guides available, with 7 that are in development. And by simply going to the website and going to the appropriate location, if you click on the reagent guide, in this case, I've shown here oxidation of aldehyde, oxidation to aldehydes and ketones from different types of alcohols. What it'll do is when we double click on this, it'll take you to a Venn diagram that describes the greenest possible conditions for this type of transformation. It'll also give you useful information such as reaction mechanism. It'll give you hot links to literature showing actual, in several cases, scale up procedures for these conditions. Incredible valuable information that is there. I know myself, I always go, whenever I start a new project, I go to these and look and say, okay, make sure that have the best conditions. And certainly for academia, new students, this is a great place to go to see what the pharmaceutical industry sees as important areas of chemistry. So my key focus today is to talk about the early days of the ACS Pharmaceutical Roundtable. As mentioned, right now we're at 37 strong member companies doing great work in sustainable chemistry and engineering. And of course, as one might gather, Rome wasn't built in a day. This had to start somewhere, and really it actually started 16 years ago. It was back in 2004. And so really a key event happened there, and I'm giving a lot of credit here, enormous amount of credit here, to the early leaders of the Pharmaceutical Roundtable. Buzz Q was an upper level executive at Pfizer who retired, and Buzz, I think when he retired from Pfizer, really had a strong itch to continue in working in green chemistry. And so he had an idea, along with Paul Anastas, as many will know, is one of the true founding fathers of green chemistry. It's the third bullet point here. They got together in 2004 and discussed the possibility of forming a pharmaceutical community of practice to address common green chemistry's issues and opportunities, with the mindset of establishing a pre-competitive consortia. They also interacted with a key executive at Lilly, Chick Vladichek, had a lot of discussions, and then they met at a conference later in 2004, Sustainable Chemistry and Engineering workshop in Philadelphia, and that further led to more distillation of their concept for founding the roundtable. And so in 2005, there's the inaugural kickoff meeting for the Pharmaceutical Roundtable, and have some quotes in here. And I thought what was really interesting was the last sentence in the quote here, which I'll read. It says, "While interest and enthusiasm from industry remains high, it should be noted that it is being launched at a very precarious time for the pharmaceutical industry, which faces numerous challenges in 2005. So it's very interesting that we think of challenges we have today in the pharmaceutical industry in 2020. Back in 2005, there were challenges just as large, if not larger. One key event that did happen shortly after the roundtable founding in 2005, and this basically kickoff meeting is largely recognized as the founding of the roundtable. Julie Manley was certainly someone that was instrumental over the first decade. She was hired in by the ACS GCI, and she helped greatly on recruitment of member companies and coordination of all the activities were needed. A big point here is that the roundtable agreed very early on to limit work to pre-competitive, non-competitive items. And what that means is the roundtable agreed with a code of conduct that there would not be any treading on other companies' IP. But at the time, this was a very groundbreaking concept, because to be honest, the pharma companies weren't used to working closely together at this point in history. So this was a really, really groundbreaking concept in 2004, 2005. And sort of our founding fathers have to be highly acknowledged and praised for the outstanding work they did to position us for success today. So what are some of the guiding principles for pre-competitive collaborations? Really, there are 4 key bullet points that are needed for this to be successful. And that's that companies in the same market, or principles here, the companies in the same market can work collaboratively. Those same companies can simultaneously compete for market share, and they explore new ideas to advance scientific knowledge, and very importantly, have respect for each other's IP. Really need all 3 of the, or all 4 of these items to come together to form a community of trust to enable growth. I show here a picture of a football field on the right, and the important concept there is that if you think of a company's IP, that's like being in the red zone. But the reality is there's a lot of, and that's the area you don't, in a pre-competitive consortium, you don't work in, but there's a lot of other space on the football field where you can work together that's non-proprietary, and that's what the roundtable takes advantage of. Big early decision here was to establish the Pharma Roundtable as nonprofit, which allows for funding of much research, grants, sponsorships of conferences, and so forth. Won't spend any time here. This just shows from the desk of Paul Anastas the agenda for the initial roundtable meeting, which was held in early 2005, starting everything off. And we'll also acknowledge, we'll thank the Boston Red Sox as well. You note this is an authentic message from Paul Anastas that basically mentions a lot of nonstop celebrating after the Red Sox won the World Series in 2004, and that after about a month of celebration, he says, you know, we ought to get on to the round table. Let's get that going. And to be honest, I was during that period, I was celebrating just like Paul was as well, because it was great to see Boston do so well. So one of the key pieces to success of the Pharmaceutical Roundtable, it's really about the people who contribute and participate. And this group here is the list of co-chairs that have led the Pharmaceutical Roundtable since day one. We're up to our 18th co-chair now, which is Louis Durazio from AstraZeneca, and he leads the roundtable right now along with Paul Richardson from Pfizer. Shown here also, if you look, our first 2 co-chairs were Buzz Q from Pfizer and Chick Vladicek from Lilly. And so how the roundtable is structured is we have 2 co-chairs at any given point in time, but it rotates. So there's a fresh election of a new co-chair every year. These people are all key leaders, not only within their own companies in sustainable chemistry and engineering, but also they're globally recognized. And really, this dedicated group is what makes the roundtable tick at a very, very high level. And even when people have when they finish with their co-chair responsibilities, if you look at over the past decade, all the people in the past decade that were former co-chairs are still very active in the roundtable and key ambassadors. So this is just a great group of scientists, individuals, leaders in their companies that are helping move the Pharmaceutical Roundtable and promoting tremendous messages of the importance of sustainable chemistry and engineering. If you look at the roundtable growth, as mentioned, back in 2005, started out with 3 companies in GCI, and it quickly expanded within a 5-year period up to 16 companies in 2010. And but the growth trajectory wasn't always linear. If you note here, we had, you know, a roughly 4 to 5 year period where it was a little bit of rocky road, at least in terms of membership, where we actually lost revenue or lost membership, do some mergers and acquisitions, and in other events. And so we really had a very flat to growth loss for about a 4 or 5 year period. But note now, since 2016, we've had a very, very rapid trajectory of growth. Now we're up to the 37 company level. A couple key things that have happened. One, we did change our business plan in 2017, when effective at the beginning of 2018, where we now allowed other companies with focus on green chemistry, sustainable chemistry and engineer, that were related to the pharmaceutical industry to join. We got a great uptick in membership from that. We've also had a very significant amount of peptide companies join the roundtable as well. And interestingly, we also see this medium molecules, large molecule area as a further growth driver, and expect that potentially we could add many more companies in the oligonucleotide sector in the upcoming years. So one of the most important things that the Pharmaceutical Roundtable did in the early days was publish a perspectives article. So within 2 years of the official formation, all the member companies at that time published an article in Green Chemistry, which has now been cited 882 times, that really just laid out the principles of what the roundtable was all about and focused on 12 key research areas. This also was the foundation for numerous research grants. And there was actually a reboot of this manuscript that was published in 2018, which took a look and actually added a few areas that the roundtable thought was important. And so this just highlights the most recent, the 2018 research area. I'm not going to read through all these, but just highlight a couple of them. Big focus on inexpensive, sustainable metals and methodologies. And I believe you'll hear very good you'll hear very good talks coming up in this symposium related to this topic of the great work going on there. But just the concept is, if you look at the periodic table on the right, the items in red or shaded in the dark orange are elements that have risk of perishing, of disappearing over the next 100 years, or possibly even less. And so need to have those replaced with non-precious metals that are safe, that are abundant. That will continue to be a critical area for years to come. Also, just highlight on the bottom, always focus on solvent replacement, trying to avoid solvents if you can, but very importantly, using the safest possible solvents and avoiding halogenated solvents if at all possible. So the Pharmaceutical Roundtable, because we're pre-competitive, a large amount of our money goes directly into research grants. And since 2007, as you'll see here, we funded numerous academic researchers, and yeah, we funded numerous academic and awarded over $2 million in grants, which have led to 65 publications with over 2,000 citations. We'll also highlight that researchers were also able to leverage, based on this work, a number of them federal funding as well. So that's probably one of the most important things that we do is fund academic research. And one of the real benefits of being nonprofit is that we can fund revenue we generate. We can basically turn that around, offer research grants, fund conferences like the Green Chemistry Conference, and do all sorts of great work with that revenue we have coming in. So just wanted to highlight very briefly, you saw this, the Pete Dunn Award. We also have an award for excellence in research development within the pharmaceutical industry. And you'll see here our initial award winners. Pete Dunn was our initial. Last year, Frank Gupton, which we'll hear speak later in the proceedings, received an award largely for his work in the Medicines for All initiative. And in about 15 minutes, actually maybe less, in about 10 minutes, the 2020 award winner will be announced. So please stay tuned for that. And we also are already looking ahead towards the award next year. I'm going to move through this pretty quickly because time is short. But one of the best things the Pharmaceutical Roundtable developed was a common material mass metric, a process mass intensity defined as total kgs of material used in a process for kg of API. What we found from our benchmarking study was an average commercial process would use approximately 170 kilos of material for each kilogram of product, and the distribution was about 60% of that waste was from solvent, about 30% from water. And so this was a benchmarking we published, and just want to show here the roundtable is also now looking at other therapeutic classes. As mentioned, small molecule, a PMI of about 170. Some of the other areas we benchmarked in biopharmaceutics, we've looked at a study with monoclonal antibodies. The waste generated there is approximately 7,700 kilos of waste per kg of product, and in oligonucleotides we're up to 7,000. So it leads to a good question: is where are areas the roundtable will invest in the future? As you see, outside of small molecule, there's very, very significant waste being generated in industry. Real quickly, one of the best things about being involved in the roundtable is you get to see what's coming in the future. There's always a good look ahead, or planning ahead for what's the next 5-year or so horizon look like. And so that's one of the great benefits of being a member of the roundtable, is that you get to participate in these key initiatives and really be at the forefront for what's changing in the industry. So as mentioned, the ACS Pharmaceutical Roundtable is not just regional; it is global. In the last year, we had one of our 3 meetings we have every year held in China for the first time, hosted by both at Pharmaron, at ASIMChem in China. You see a photo there of the group that was there at the Great Wall. Extraordinarily well received. And this was perhaps one of our top meetings we had, where we had quite a few interactions with the Chinese the Chinese officials, members of companies, discussion of issues environmentally in China and steps that could be taken to improve the situation. Pharmaron hosted a major scientific seminar that we all participated in as well. So in the future, we hope to have further such meetings in different locations in the world. I will mention that how we are currently structured, we do have every year, as long as I can remember, one of our annual meetings is always held the same week, concurrent with the annual Green Chemistry and Engineering Conference, and that I believe will continue. Will continue in the future. So in conclusion, the Pharmaceutical Roundtable is shown to provide, it does provide leadership and influence, not just in industry, but in academia and society in general. Great tools are made available publicly, and I didn't talk about too many of them. Show the Reagent Guide. They're there for all. Key decision made by the early founding fathers was to have us be set up as a pre-competitive nonprofit group, and that was really critical, and something that would consider other groups deciding whatever the cause being. I think having a non-for-profit status is absolutely critical, because there's just so much more you can do when you're in that situation. And also highlight that the roundtable is not static; it's always evolving its mission since 2005. And when it started out, it was more or less exclusively a small molecule-focused organization, and more or less a brick and mortar. It evolved since 2005, not just to be small molecule, or not just to be internal to companies, but focus on the whole value chain, supply external to our companies, contract research. And now there's been great expansion outside of small molecule into large molecule, into other modalities such as peptides and oligonucleotides. And so that's just critically important that the roundtable continues to grow. And finally, I'd like to point out, there's an open call new members here. We're looking for more to join us. So please, contact the ACS if you have interest. Finally, on the acknowledgements, there are two people that really stand out above and beyond on their contributions to the roundtable over the years. The first I'll highlight is Julie Manley, who was with the roundtable for about the first decade, helping getting the roundtable going, doing a lot of the administrative work, involved on several of the leading initiatives that helped really get the group out of the gate. Second person is Ismer Martinez. Ismer Martinez has been involved with the roundtable now for at least 4 or 5 years. It really has led us through this great period of growth, unprecedented period of growth, where I think when Ismer joined, we were at about 13 or maybe 14 companies, and now, we're up in the 36, 37 range, and a lot of that has been the great work of Ismer promoting our message and helping manage all these different teams we have going on. So really thank her. Of course, thank you, more importantly, for your support, and I'd be happy to take any questions you may have.
Thank you, Mike, for the great presentation. And we do have time for some questions. So the first one that we have is, where do you see the role of excipients suppliers in sustainability? And I guess I can broader, do you see a role for the Pharmaceutical Roundtable to include, drug product formulation?
Yeah, Juan, that is an absolute great question. I would say unambiguously yes on the drug product. That's a source of significant waste, and I know that there's a lot of effort goes to producing API, and always heard that extra material gets made because 20 to 30% gets lost in the pipe. So that's an area that's totally ripe for a roundtable sub-team, is drug product formulation, and I believe we should consider forming a team in that area. Regarding excipients, yeah, I think there is some miles on the tire there. That would fit in well in our affiliate status, and I think it's something we should take a look at.
Thank you, Mike. Another question from Josephine is, how does the biochemical, biological technologies compare to the small molecules or chemical drug technology environmental impact regarding waste, air, and emissions?
Okay. So regarding waste, they're very different processes in large molecule than small molecule. These generally tend to be recombinant technologies, which actually in general tend to use very little solvent, perhaps with the exception in chromatography for purification, but they use enormous volumes of aqueous, basically, which is actually a very big problem. As shown on the other slide, there could be 8,000 kilos of water per kilo of API, and in some cases it can be much higher depending on your class. So that's an important, a very important area to focus on. We do have a roundtable sub team that's active in that area. The other are the modalities, the larger molecules as well, the synthetic peptide and oligonucleotides.
Yep.
Those are areas that could generate waste up to 10,000 kilos of organic waste per kilo of API. That's certainly a major focus in the future, is to improve that.
That's great, actually, Mike. We have another question from Jared that addressed the first two questions. The Pharma Roundtable has grown a lot in size. Is there a limit of the number of members that we can hold, or do you expect us more different type of modality and businesses are interested to join? You will have to, or we will have to create new subgroups of that.
That's an excellent question about how to manage growth, and I think that I don't have an easy answer for that. Personally, I'd love to see the roundtable keep growing, but at a certain point, the organization could get too large, and that could be hard to manage. So I think in the future, we don't want to exclude anybody, there may be a need to have the split off into some sub roundtables, perhaps. You know, for example, does the biologics and peptides split off into their own areas if they have critical mass? Things to consider like that. And I think that's just something collectively I think we're going to have to, I think, discuss, you know, the leaders of the current roundtable in the upcoming year or so.
I agree. And that's a great answer. So, and then one final question from Bikren. Did you anticipate the changes in pre-competitive guidelines? Any changes in the post-COVID period, COVID-19 period?
Is the question related to the pre-competitive rules, or I didn't quite...
looks like in terms of the pre-competitive rules, do you expect any changes on that?
I personally, I would say probably not. I think the roundtable, I think mission would stay the same and independent. I think independent of the virus. I don't think that would have an impact on us one way or the other.
Right. As I may add, I mean, we have seen in the industry a lot of collaboration between pharma to really find solutions for the current situation. And certainly the Pharma Roundtable, we're doing that, right? By working in pre-competitive, certainly we will try to advance that type of collaboration. So.
Absolutely.
Okay, Mike, well, thank you. I think we're just in time. And thank you again for the great presentation. And now, we would like to move to the next agenda item, which is the presentation of the Peter J. Dunn Award by the ACS Green Chemistry Institute Pharmaceutical Roundtable co-chairs, Frank Gupton and Paul Richardson. I think we're going to start with Frank and follow with Paul.
Thank you, Juan. Can everybody hear me?
Yes.
Okay, perfect. Okay, thank you everyone for attending today. So my name is Frank Gupton. I'm from Boehringer Ingelheim. Unfortunately, I had technical difficulties with my webcam, so this is my photo, this is Paul, and this is Isamir, who also contributed to the slides. I'm very thrilled to present today, co-jointly with the other co-chair, Paul Richardson from Pfizer, the 2020 Peter J. Dunn Award on behalf of the ACS Green Chemistry Institute Pharmaceutical Roundtable, and specifically the Roundtable Awards team. We already heard a little bit about the award. The award, oops, I'm actually on the wrong slide, I'm sorry. The award was established in 2016 by the Roundtable in honor of Peter J. Dunn of Pfizer. It recognizes excellence in research and development and also excellence in execution of pharmaceutical green chemistry. The winners have to demonstrate compelling environmental, safety, and efficiency improvements over current technologies. So Pete Dunn, whose picture is shown here, and after whom the award is named, was recognized in 2016 for his leadership of Pfizer's green chemistry program and his accomplishments in a greener total synthesis of sildenafil citrate, or also known as Viagra. And this kicked off the first Pete Dunn Award. Afterwards, in 2017, Professors Bruce Lipschutz and Sachin Handa won the award for development of a newly engineered catalyst for sustainable surfactant chemistry in water. And this really allowed the reduction of the amount of palladium needed to carry out cross-coupling reactions for pharmaceutical applications. In 2019, Professor Frank Gupton, who's also a speaker later today, won the award for increasing global access to the high-volume HIV drug nevirapine through process intensification. Before I move on to the winner of this year, I would like to note that the 2021 nominations, submissions, will open in the fall of 2020. And I invite you to follow this link and make a submission. Now, the winner is, I apologize for having shown the slide before, it's Dan Bailey, colleague from Takeda. And his photo is shown here. He won the award for a submission titled Beyond Organic Synthesis, Synthesis of a 5-HT4 receptor agonist in water. Of course, we'll hear more about it in detail during his presentation following this introduction. Here is a picture of the actual award plaque, so you can see it. The slides will be distributed later, I assume, so you can follow these links for announcements in chemical and engineering news and also in the ACS Green Chemistry Nexus blog. And now a little bit to the award. Already mentioned the title. This target drug is shown here, this TAK-954. It's assembled from these 5 building blocks. And the work of Dan is the very first application of aqueous surfactant chemistry to an entire manufacturing process of a complex active pharmaceutical ingredient. During this work and process optimization, Dan and his team developed 2 novel transformations to occur in surfactant modified aqueous media. So the innovative novel transformations, namely a reductive amination and an aliphatic alcohol oxidation. By implementing these innovations, they were able to reduce the step count of the overall process from these commodity type starting materials from 7 to 5, and they were able to reduce manufacturing waste as indicated by complete E-factor or PMI -1 by almost 2/3, from 350 kilograms of waste per kilogram of produced drug to 110. Part of this waste reduction was 90% reduction of organic solvents, and surprisingly also a reduction of water waste. And with this, I will hand over to my co-chair, Paul Richardson from Pfizer, who will introduce Dan Bailey.
Thanks, Frank. Morning, everybody, afternoon, wherever you may be. It's my great pleasure to introduce our award recipient, Dan Bailey, this morning. I think we changed the slide. Dan received a dual BA in chemistry and anthropology from Brown University in 2009. I'm very hopeful that today we're going to hear more in his presentation about chemistry than anthropology, because he'll definitely lose me if he talks about anthropology. During his studies, he was awarded the Clapp Prize for Outstanding Undergraduate Honours Thesis in Chemistry, developing a general synthesis for the methylomycin furans with Professor Jason Sello. That work, I believe, was published in All Collect in 2009. Since then, he's been working in process chemistry at Takeda in Boston, supporting preclinical to late-stage development projects, and he leads efforts there to track and improve the efficiency of sustainability of Takeda's API manufacturing processes. In addition, with his activities over the last couple of years with the Roundtable, Dan has led up the chemistry and water team there. And I could say there's quite a few exciting developments we're going to see in that space in the not too distant future, with studies ongoing in terms of benchmarking efforts, as well as studies in terms of the grant process to look at downstream processing of aqueous processes. It's my great pleasure to congratulate and introduce Dan, and look forward to hearing his presentation today on Beyond Organic Solvents, synthesis of a 5-HT4 receptor agonist in water. Dan.
Okay, thank you very much, Frank. Thank you very much, Paul. Can everybody see me okay here? Is the audio all right?
Yes.
Okay, perfect. So yeah, I'm thrilled to be accepting the Peter Dunn Award this year. Actually got the award right here. I've had it for a couple of days, but we can pretend that it was just handed to me now. So I want to make sure I mention the Pete Dunn Award sub-team of the Pharmaceutical Roundtable. I know they've done a lot of work and put a lot of careful thought and consideration into thinking about the significance of the award this year, and also in reviewing all of the nominations. So I'm honored that they selected my work. I'm also just happy that this award exists. I think it's a great way to encourage investments in sustainability in pharma. And really, I think sustainability is probably one of the biggest challenges facing the pharmaceutical industry today, and that's, I guess, true of a lot of industries these days. But there's really a need to continue to provide life-changing medicines to patients, while also ensuring that patients and workers and the communities where we operate have access to a future that's safe and healthy and just and ecologically balanced. And to think that my work may be contributing to that goal, even in a small way, makes me really proud and very pleased. So, of course, I didn't do this work on my own. I need to acknowledge my employer, Takeda, for investing in this work and for investing in green chemistry in general. And also, right up front here, I want to make sure I thank my colleague Dave Leahy, who nominated me for the award. He's been really a great supporter and advocate of this work from the outset. When we're doing this work, whenever we ran into roadblocks, Dave was always there with advice and ideas and encouragement. And of course, a number of other people also contributed to this work in a technical capacity, and I'll tell you a little bit more about their contributions in the talk in a minute here. But I do want to also mention that it's been about 2 years since I've gotten involved with the Pharmaceutical Roundtable group, and it's really been a pleasure getting to know some of the people in the group and learning more about the work that they do and getting involved in some of the Roundtable's initiatives. And the Roundtable really does, high quality and very important work. So I'm glad that it exists as a resource for the industry. Okay, so let's get into the technical part of the talk. I think that's what we're all here for, right? So today I'm going to tell you about some work that was done at Takeda to adapt one of our drug manufacturing processes to be run almost entirely in water. And that includes reactions, but also workups and isolations. And I should mention that we will be publishing this work imminently. So I'm excited to give you a little preview today, but if you are interested in the details, keep an eye out for the publication. So first, why are we interested in doing drug manufacturing in water? Well, the pharmaceutical industry kind of has a solvent problem. So if we take a look at the materials that we use to manufacture active pharmaceutical ingredients across the entire industry, the majority of those materials, so at least 56%, some estimates are as high as 60% or higher, are organic solvents. So that's a lot of solvent, and most of that solvent ends up as waste, of course. But maybe more concerning is that a lot of the solvents that we use are hazardous. So they're toxic, they're hazardous to human health. Many of them are flammable, so they pose a risk to worker safety. And then many of them are environmental pollutants, so they're volatile. And whenever we use them, a certain amount escapes into the atmosphere where it can contribute to smog formation and cause other problems. And then, of course, most of the solvents that we use are derived from petroleum, so they're non-renewable. So clearly there's a problem here. What can we do about it? Well, a lot of people who thought about this problem have concluded that water might be a promising alternative to organic solvents. And that includes Roger Sheldon, who's sort of been a leader in green chemistry for many years. And in 2017, he wrote, "The best solvent is no solvent, but if a solvent is needed, then water has much to offer. It's non-toxic, non-flammable, abundantly available, and inexpensive." So clearly there's some benefits to using water. Maybe there's some drawbacks as well, and the big one is solubility. So a lot of organic reaction components just aren't that soluble in water. And one way to deal with that issue is by using surfactants. So surfactants in water form these structures called micelles. So this is sort of a cartoon cutaway of a micelle, where there's a lipophilic core surrounded by a hydrophilic shell. And the idea is that you can use these micelle structures sort of as little nanoreactors to do chemistry. So you can imagine your organic reaction components would migrate into the lipophilic interior of the micelle where they could react and make products. So can we use just any surfactant to do chemistry in water? Sort of, but some certainly work better than others. And one of the most commonly used one is TPGS 750M, which is shown here. This is a surfactant that was specifically designed for doing chemistry in water. It's derived from vitamin E, so the idea is that it's benign by design. It was developed by the Lipschitz group at UCSB. And this is a general surfactant for doing chemistry in water. So a lot of different chemical transformations will work in water using this surfactant. And if you are interested in learning more about molecular chemistry or chemistry in water, there's a couple of talks coming up, I believe, on Monday at the conference that you should check out. So Bruce Lipschitz will be giving the keynote address, so you won't want to miss that. And then later in the afternoon on Monday, there's actually an entire session devoted to chemistry in water. So be sure to check that out. So the next question then is, can we use molecular media or water to do large-scale drug manufacturing? And Novartis has made a lot of progress in this area in the last couple of years. And one of their key findings has been that use of a small amount of an organic co-solvent actually enables scale-up of molecular media. So this is an SNAR that was developed at Novartis. You can see when they're just using surfactant and water without any co-solvent, they get this kind of gumball here. At 10% THF, things start looking better, and then at 15% THF, they get this nice stable emulsion that they can scale up, largely independent of mixing effects. So we were inspired at Takeda by some of the recent work in the area of chemistry and water. We wanted to see if we could apply some of these ideas to our portfolio. So we decided to give it a shot, and we chose this project, the TAC954 program. This is the target molecule here, TAC954. It's a 5-HT4 receptor agonist, and the current indication is postoperative GI dysfunction. So when we began this project, this is how we were making this molecule using organic solvents. So the synthesis begins with the condensation of the preformed bisulfite addict of isobutyraldehyde with this diamine, followed by cyclization to get this benzimidazole intermediate. We then do a direct amidation of the methyl ester to install this first piperidine ring, followed by a Bhakti protection. And we then installed a second piperidine ring using reductive amination, followed by a second Bhakti protection. And then finally, we installed the methyl carbamate functionality of TAC954 using methylchloroformate. And then there's an acetonitrile cooling recrystallization at the end here. So the overall yield for this sequence is 35%, not too bad. But if we take a look at the cumulative process mass intensity, so for those of you who aren't familiar with PMI as a metric, it's essentially just a measure of the amount of stuff that it takes to make 1 kilo of product. So if we work through this sequence here, we'll need 350 kilos of stuff to make 1 kilo of 954. And that's a relatively poor number. That's fairly high. And that indicates relatively poor process efficiency. So there's definitely room for improvement here. We're also using 5 separate organic solvents in this sequence, so toluene, trifluoroethanol, NMP, dichloromethane, and acetonitrile. And some of these are solvents that are relatively hazardous, and we really prefer to avoid. So you may have also noticed that there's sort of a synthetic inefficiency in this route. So we're bringing in this second piperidine ring with a Bach group, a T-butyl carbamate, already installed. Then we're immediately removing that Bach group and installing a different carbamate, so methyl carbamate. And aside from that inefficient protection deprotection strategy, we also form significant amounts of this bis carbamate impurity, which we can remove during extractive workup, but really it's something we prefer to avoid forming in the first place. So there's definitely an opportunity here for a more efficient route. And you can imagine that more efficient route might look something like this, where the first 3 steps, the benzimidazole cyclization, the amide bond formation, and the Bhakti protection are essentially the same. But then in the final step here, we're bringing in this aldehyde fragment with the desired methyl carbamate already installed, which gives us direct access to TAC954 and cuts 2 steps out of the sequence. Unfortunately, this aldehyde fragment is not readily available, which is one of the reasons we weren't using it in the initial route. So we have to think about how we might make that. And you can imagine making that from this hydroxymethylpiperidine starting material, installing the methyl carbamate, and then oxidizing the alcohol to the aldehyde. So this is the route that we decided to pursue. And now I'll walk you through some of the work that we did on each step to adapt this chemistry to water. So the first step is the benzimidazole cyclization. As a reminder, we're preforming the bisulfite addict of isobutyraldehyde and then condensing that with this diamine followed by cyclization to give the benzimidazole product. And when we initially tried these conditions in water, they worked. We got product. But unfortunately, we also got a significant amount of these bis addition impurities, which form as a result of either the aldehyde or the bisulfite addict adding to the diamine twice before cyclizing. So a lot of the work that we did in this step focused on thinking about how we could suppress the formation of these bis addition impurities. And in thinking about that, we thought about some of the factors that contributed to the formation. The first thing we found is that the free aldehyde exclusively gives bis addition. So if there's any residual aldehyde hanging around, we're going to form these byproducts. So in order to control that, we optimized the amount of sodium bisulfite that we're using to form our bisulfite addict. And since this is an equilibrium process in water, we did need an excess of sodium bisulfite in order to push that equilibrium completely to the addict. The next thing we looked at is solubility. So the diamine starting material is not fully soluble at pH 7 in water. And that means that we don't have good control over the relative amounts of diamine and bisulfite addicts that are available to react at any given point during the reaction. So in fact, because the bisulfite addict is fully soluble in water, we actually have an excess of bisulfite addict throughout the reaction, which is exactly the situation we want to avoid forming these impurities. So in order to solubilize this diamine starting material, we took advantage of the fact that we have this acid functionality. So we can use a pH adjustment to control solubility in water. So if we use hydroxide and adjust the pH to greater than 12, we can get full dissolution of this material. The next issue is sort of a related problem. So we had initially been adding the bisulfite addict in a single portion, which again meant that we didn't have tight control over the relative amounts of diamine and bisulfite addict that were available to react during the reaction. So instead, we implemented a controlled addition of the bisulfite addict over 3 hours in order to maintain an excess of the diamine. So with those three changes, we were able to suppress the formation of those bis addition impurities to essentially undetectable levels. And now that we've formed the product cleanly, we need to think about how to isolate it. So throughout this process, it was important to us that we implement direct isolations, ideally crystallizations, from the reaction mixtures. So the goal here was really to drive down the amount of organic solvent we're using. So we didn't want to be adding additional organic solvent during workups and isolation. So in this case, again, we could take advantage of the fact that we still have this acid functionality in our product. We can use a pH adjustment again to isolate it. So if we adjust the pH to less than 5 using HCl, we get crystallization of the product. We can isolate it by filtration in 80% yield. PMI for this step is 24.5. So next step is the amide bond formation. So first things first, we needed an activating agent. So we screened a bunch of different activating agents, things that form acid chlorides, mixed anhydrides, activated esters, various other activated species, sort of an alphabet soup of your acronym coupling reagents. But ultimately, we ended up settling on these conditions here, so TCFH and N-methylimidazole. And these conditions work by forming an activated acyl-imidazolium species, which has a similar reactivity to an acid chloride. These conditions were developed a couple of years ago by chemists at BMS, and they originally identified acetonitrile as the optimal solvent. We found that these conditions work equally well in water, which might be a little surprising since TCFH and the activated species look like they wouldn't be particularly water stable. But as long as we add TCFH last, and as long as we have our amine nucleophile available to capture this activated species, the chemistry works well. And it actually works almost too well. So this is a nearly instantaneous reaction. It's done within a couple minutes. And the issue is that our starting materials are fully soluble under the reaction condition, but our product is poorly soluble in water. So that means over the course of the reaction, we're building up a tremendous amount of supersaturation of our product, which tends to lead to product oiling and reactor fouling. It looks pretty nasty. It looks like this. So a lot of the work that we did here involves thinking about how to mitigate this oiling issue and get a nice controlled reactive crystallization. So the first thing we did is we added THF as a co-solvent. The idea here was just to modestly improve the solubility of our amide product and reduce the amount of supersaturation that builds up. Next thing we did was we added the TCFH in 3 portions. So again, the idea was to slow the reaction down a little bit and reduce the amount of supersaturation of our product that was building up. And then finally, we found that the surfactant itself seemed to actually be inhibiting the crystallization of this product. And since our starting materials were fully soluble under the reaction conditions, we weren't really doing micro chemistry anyways. So we ended up removing the surfactant. And with those changes, we were able to get a nice filterable slurry here at the end of the reaction. We could just filter it directly, isolate the product in 91% yield. PMI for this step is 9.2. So next up is epoxy protection. Not a lot too interesting going on here. This is the kind of chemistry you'd expect would work in water, and it does. So we're using HCl at 50 degrees. The reaction's done in about an hour. Both our starting material and our product are fully soluble in water at low pH. So we can use another pH adjustment to isolate the product. If we adjust the pH to greater than 12 with a slow addition of sodium hydroxide, we can crystallize the product in 88% yield with a PMI of 24. So just to reorient you quickly, so this is the first 3 steps of the sequence that we've made it through. The chemistry is more or less the same as the chemistry we were using in the enabling route. We've just changed to different conditions in order to accommodate the aqueous conditions of each reaction. But the remainder of the route is sort of more uncharted territory. So first, we need to prepare this aldehyde fragment, and then we need to do a reductive amination. And at least when we did this work, there wasn't a lot of literature precedent out there for doing reductive aminations in water. And there also wasn't a ton of precedent for doing oxidations purely in water as well. So let's start by talking about preparation of that aldehyde fragment. So we begin with hydroxymethylpiperidine here, install this methyl carbamate functionality using methyl chloroformate. This works well in water, it's clean, it's fast. The carbamate here is an oil, so we're not isolating it, we're just carrying the reaction solution on to the next step, which is the oxidation. So the first thing we tried here was your standard tempo bleach oxidation conditions, and these worked. You know, we got some products. Unfortunately, we also got a significant amount of over-oxidation to the carboxylic acid, as well as some residual unreacted alcohol starting material. So we went back to the drawing board and screened a few other conditions, and we ended up settling on these conditions. This is also a tempo-catalyzed oxidation, but we're using phenyliodide diacetate as our stoichiometric oxidant. And these conditions give us full conversion to the aldehyde. Most importantly, we don't see any overoxidation, even after prolonged stir out of up to 3 days. We do need surfactant and co-solvent here, mainly to solubilize the phenyliodide diacetate oxidant. So here we are, we're finally doing some micellar chemistry. And now that we've formed our aldehyde, we need to think about how we're going to isolate it. So this is also an oil, and we wanted to use this material as a registrational starting material in our final API forming step. So for us, that means we want to have good control over the physical properties of this material and its stability. So we chose to derivatize the aldehyde so that we could isolate it as a crystalline solid. So we formed the bisulfite adduct, which allowed us to isolate the material as a crystalline solid. As an added bonus, bisulfite adducts are known to be more stable than free aldehydes. But there were some challenges in isolating this material, mainly stemming from the fact that the bisulfite adduct is very soluble in water. And since we're operating in water, really we had no choice but to use some sort of organic anti-solvent in order to crystallize it. We chose ethanol for that purpose. But the other issue here is around isolated purity. So we do tend to co-precipitate some of the reaction byproducts with this material under the crystallization conditions. So we get a little bit of residual metabisulfite and then a little bit of residual acetate salts coming from the phenyliodide diacetate. But we can tolerate those impurities well in the next step. So something we can live with. So here's the complete 3-step sequence to the bisulfite adduct fragment. We're isolating it in about 50% yield and 70 to 80% weight assay. Again, those impurities are well tolerated in the next step. PMI for this entire sequence is 17.9. Okay, so we're in the home stretch here, the last step, the reductive amination. So first things first, we needed a reductant for this transformation. We needed something that would be water stable. So we ended up choosing picoline borane for that purpose. And there's some literature precedent for using picoline borane to do reductive aminations. We realized pretty early on that we were going to need both surfactant and co-solvent in this step, both to get a workable reaction mixture to help solubilize our amine starting material, but also to get full conversion. So having that micellar environment, that lipophilic environment within the micelles really helps to overcome the thermodynamic barrier to imine formation in an aqueous environment. But the big question here was, can we directly use the bisulfite adduct in this step, or do we need to regenerate the parent aldehyde first? So the first thing we looked at doing was separately regenerating the parent aldehyde. So we did that in hydroxide water. We could get full conversion back to the parent aldehyde. And then if we use that aldehyde solution in the reductive amination, as long as we had 1.5 equivalents of our reductant, 1.5 equivalents of the aldehyde, we could get full conversion to TAC 954, and we didn't see any reduction of the aldehyde. So that was very promising. The next thing we looked at was directly using the bisulfite adduct in the reductive amination in the presence of exogenous base, so sodium acetate. The idea here is that we could reform the free aldehyde in situ and allow the reaction to progress. And there's also some literature precedent for this strategy. This also worked. But then over the course of optimizing the amount of base we were using in this reaction, we ended up trying a reaction without any base. And that also worked, which was a little surprising at first, but maybe not that surprising if you think about it. So in water, bisulfite adducts and free aldehydes are in equilibrium. And even if there's only a small amount of aldehyde present at any given time, Le Chatelier's principle tells us we'll slowly siphon off the bisulfite adduct and form product. And that seems to be what happens. So that led us to a pretty simple reaction protocol. We essentially just mix everything together, heat it up to 60. It's done in about 8 hours. Then we implemented a reverse isolation, mainly to avoid some oiling issues in this step as well. So we're adding our reaction solution over 3 hours to a seeded solution of hydroxide and water. That gives us a nice controlled crystallization of our product, which we can isolate by filtration in 91% yield. PMI for this step is 14.7. So we are isolating the product as a hydrate, which is not surprising since we're operating in water. Unfortunately, the desired development form for this program is an anhydrate. And essentially, it's not possible to isolate the anhydrate from aqueous conditions. So we needed sort of a non-aqueous recrystallization step in order to convert the hydrate to the anhydrate. We chose to use the acetonitrile cooling recrystallization from the enabling route for that purpose, so we can convert the hydrate to the anhydrate. This is really the only step in the process that's done in not in bulk aqueous media, and it's essentially unavoidable. So that's it. We've made it through the whole route. So this is 6 steps, almost entirely in aqueous media. 5 direct isolations from aqueous media. So how does this route compare to the enabling route? So I actually have some updated results here. We've run this on scale and demonstrated it. And if we just take a look at the 4 linear steps, the linear sequence rather, which is kind of the direct comparison to the enabling route, because in the enabling route we were not preparing this aldehyde fragment. So the enabling route overall yield is 35%, and in the water route we're at 56%. If we take a look at the cumulative PMI, the enabling route was 350, and for the water route we're down at 70, which is about an 80% reduction in the amount of materials that we're using to manufacture this product. If we look at the solvent PMI, so we're at 223 in the enabling route, and we're down at 15 in the water route, so that's about a 90% reduction in solvent use. Maybe not that surprising since we're using water as our bulk reaction and isolation medium. But if we take a look at the water PMI, in the enabling route it was 106, and in the water route we're down at 49. So that's about a 50% reduction in the amount of water that we're using here, which is maybe a little more surprising, but I think we often forget that even when we are using organic solvents, we often use a lot of water as well, particularly in workups and isolation. And then most importantly, the API quality, so the purity of the API isolated from these 2 routes is more or less comparable. So some lessons learned. When operating in water, pH can provide a solubility handle, allowing for direct isolation. So this is a trick we use over and over again in this process. And because a lot of pharmaceutically relevant compounds have a lot of heteroatoms and at least 1 or more acidic or basic centers, there may be some generality to this approach. In some cases, water can function as a solvent without any additives. So in the first 3 steps in the sequence, we were sort of truly using water as a solvent. We weren't really using any solubilizing additives. And I think as chemists, we often sort of overlook water as a potential solvent and assume it won't work well. It may work in more cases than we might think. And more chemistry works in water than you might expect. So again, as chemists, we're often trained to avoid water in our chemistry, particularly in reactive chemistry. But in this route, we use water for some reactive chemistry. So the TCFH amidation, for instance, using methylchloroformate in water. So sometimes even reactive chemistry works in water, and sometimes it's worth just giving it a try. Swapping organic solvent for water is no substitute for good process design. So water itself is not a silver bullet. If we're looking to develop the most sustainable processes possible, we need to make sure we're also implementing the most efficient route possible and optimizing each step to ensure it's as efficient as possible. So a lot of the work that we did on this route involved that sort of process design work in addition to just swapping over to aqueous conditions. Okay, so all of the work that I've showed you today was pretty much done by these 2 guys. So Ed Helbling and Amide Mankar. They're both Northeastern University co-op students who completed internships at Takeda in our group. Ed was with us for the better part of a year, and he had a hand in developing pretty much every step I've shown you today. And then Amide was with us for a semester, and his main focus was on the isolations for the first 3 steps. So really a very impressive body of work from these 2 guys as students completing internships. It was really some outstanding work. So in addition to Ed and Amide, I have a few other people I need to thank. So my line management, Matt, Dave, and Fred, for allowing me to spend some of my time doing this work, and also for providing advice and encouragement throughout. Justin Kwan is one of our staff engineers in the group, and he provided some advice on some of the isolations that we developed. Azumi Takagi is an analyst who provided analytical support for the program. And then I want to thank really everyone in the process chemistry development group at Takeda for their support and advice and encouragement throughout this work. And thank you to all of you for tuning in and listening, and I'd be happy to take a couple questions if we have time.
Thank you, Dan. And first, let me congratulate you again for winning the 2020 Pitt Dunn Award and for the great presentation, very inspiring, I'm sure, as we have been getting many questions, although, we're limited on time. One of the questions that had repeated the most is around how the water waste for these type of processes are managed. Have Takeda demonstrated this chemistry using the surfactant and water waste at larger scale, and what are the plans if you have, again, to maybe recycle or manage the waste generated from these processes?
Yeah, so that's a great question. It's kind of a complicated question. I think this is something that a lot of people are thinking about. So aqueous waste, I guess, presents us with some options for disposal beyond what we typically see in organic waste. Specifically, thermal and biological treatment can sometimes be used to degrade any organic content or residual materials that are in the aqueous waste, which allows us to treat it essentially as wastewater that can be discharged. That sort of process requires a lot of upfront work, so it really only makes economic sense if you have a very large, well-defined waste stream that you're reproducing over and over again. You have to sort of develop an individual process to treat it. That's a possibility. But also, if there is a sort of a relatively high organic content in the waste, and by relatively high, I mean even as low as 5% organic content, it often makes economic sense, at least I've been told by people who work in waste disposal, it often makes economic sense to incinerate that waste to recover the energy from that organic content. So particularly on smaller scales, when we have this kind of aqueous waste, that's often what happens to it. But it is, I think, a challenge, and it is something that people in the industry are thinking about. And we think about waste disposal of water. I think it's important that we consider advantages and drawbacks there, but it's also important to take sort of a life cycle view of using water in terms of its comparison to organic solvents. So looking at the way that organic solvents are extracted and transported and used as a comparison to the way that water is treated, purified, used, and then eventually disposed of. And I think when we do that analysis, we'll typically find that water comes out ahead. So hopefully that answers the question.
No, thank you. And again, as every time that we start with something new, like the use of this type of components and reaction, you have to figure out the end-to-end all the way from usage to disposal. So again, but
exactly.
Thank you again. Be sensitive with time. We're gonna move on to the next presentation, but let me thank you again, Dan, for the great presentation, and congratulate you again for your work. So let me make sure to introduce Professor Jeff Byers, a good friend from the Pharma Roundtable. Jeff is Associate Professor at Boston College. He was born and raised in Las Cruces, New Mexico, getting his bachelor degree from University of Washington in St. Louis, moving later to obtain his PhD in inorganic chemistry from the California Institute of Technology under the guidance of Professor John Burkott. This work was done using and developing the kinetic resolution of alpha olefins through their polymerization with chiral and antiometically enriched iconosines. After obtaining his PhD in 2007, Jeff moved to MIT, where he completed his postdoctoral position in organic chemistry under the guidance of Professor Timothy Jamieson, working on water-promoted epoxide cyclization reactions. In 2011, Jeff moved to Boston College, where he's currently an associate professor in chemistry. Jeff's research lab focuses on fundamental and applied organometallic chemistry in the areas of energy science, sustainable polymer synthesis, and organic methodology development. And as you will see, all aimed to improve the greenery of the chemical transformation that they studies. Jeff has been involved in numerous outreach activities that promote science, including being a founder of the Paper to Plastic Summer Outreach Program and founder of the National Collegiate Scholarship Association, a conglomerate whose goal is to promote education to intercollegiate competition. Jeff has been awarded the NSF Career Award for this work in the PMSE Joint Investigator Award, and is a Research Corporation Contra Scholar. Today, Jeff will be talking about the logical catalyst design for improved performance in iron-catalyzed Suzuki-Miyata cross-coupling reaction. Jeff?
Thank you very much, Juan, for the wonderful introduction. It's a pleasure to be here today. to present our chemistry in the area of iron-catalyzed cross-coupling reactions. I want to thank everyone for joining us today. BC is as you can see, I'm at my office today, and today is our second day back, and so it's been a very eventful week for us. In addition to other events that have gone on this week, we are starting up again in lab, which I'm happy to report. So today, I'd like to talk about a project that has been ongoing in my lab for a few years now, with emphasis on logical catalyst design for iron-catalyzed cross-coupling reactions. And so I think it's important to recognize the impact of cross-coupling chemistry in the pharmaceutical industry. Many of you are, I'm sure, very aware of the many examples of palladium-catalyzed cross-coupling reactions and their utilization in drug development. And here are two examples where the bonds that I've highlighted here have been made with palladium-catalyzed cross-coupling reactions. As you can see, one of the motifs that is very common for these kinds of reactions is stitching together sp2 hybridized bonds. And this is a hallmark and a really powerhouse reaction for palladium-catalyzed cross-coupling reactions. And so over the years, many have been interested in continuing to develop this kind of CC bond-forming reaction and moving from different kinds of motifs to explore the possibility of making carbon-carbon bonds between two different types of centers that do not necessarily have sp2 hybridization or sp hybridization in them. And certainly palladium-catalyzed cross-coupling reactions have made great strides in that regard, but I think a very exciting alternative to those palladium catalysts have been addressed with the development of catalysts in the first row of the transition metal series. In particular, nickel-based catalysts have evolved over time to do some very impressive reactions, and Greg Fu is at the forefront of this kind of research. Here's a very recent paper from the Fu group where they have shown that they can do alkyl sp3 hybridized cross-coupling reactions, and also these cross-coupling reactions are stereoconvergent both with the electrophile and the nucleophile to then form these very interesting and structurally complex molecules that have high diastereoselectivities and enantioselectivities. And so certainly nickel-based catalysts are very important for the development of cross-coupling reactions, not only in this traditional kind of sense, this is a Negishi type cross-coupling reaction, but also in the area of cross-electrophile cross-coupling reactions, which has seen tremendous growth over the years. However, as Mike mentioned in his talk, there's an issue associated with palladium-based catalysts as well as nickel-based catalysts, and those issues are associated with their toxicity and also availability. And so here's a nice plot that I stole from this wonderful paper, organometallics paper, that was written a few years ago, wherein this contour plot indicates not only the toxicity of the metals, but also the ligands that are associated with those metals. So most of these reactions, palladium-catalyzed and nickel-catalyzed cross-coupling reactions, involve ligands as well as the metal. And so the toxicity of those ligands also needs to be considered when thinking about the toxicity of metal-catalyzed reactions. And in this heat plot, you will see that palladium and nickel are very yellow, not quite like mercury, obviously, but certainly much more toxic than is iron, as you can see, much more green, and in this plot, relatively harmless. And so the metal, as well as the supporting ligands that are often used for these kinds of transformations, tend to be much less toxic than the nickel and palladium-based complexes that are used in cross-coupling reactions. Moreover, as Mike mentioned, there's an availability issue for these metals, and I've highlighted just the transition metal series here in a different publication than the one from the ACS, but nevertheless the same story. You can see here that nickel and palladium, as we are currently using them, will run out in 50 to 100 years. And so This chart takes into account all of the uses for these elements for industrial processes. And so we use nickel and palladium for a lot of different things, including electronics for nickel, and palladium, of course, is often used in catalytic converters. And so those major uses are preventing us, not necessarily are we going to run out of these metals for catalysis, but certainly the cost of these metals is going to continue to increase. And as you can see, the only two metals that are in the white here, other than technetium, which I don't really count because of its radioactivity, is iron and titanium. And iron is particularly impressive because if you think about the building I'm currently sitting in, it's composed mostly of iron. And so despite the fact that we use iron for a lot of different purposes, there's still plenty of iron for us to use for catalysis. And so toxicity and availability are a real good motivating factor to move from the late transition metal complexes to the middle of the periodic table and start to explore how iron-based complexes can be used in cross-coupling reactions. However, there was a problem with these kinds of reactions, and it wasn't that they didn't exist. There's plenty of evidence for iron being a useful metal for cross-coupling reactions. In fact, these two reports from Karash and Kochi predate Stille coupling reactions that are catalyzed by palladium, which happened in the late 1970s. And so many people point to this Kochi work as really the beginning of iron catalyzed cross-coupling reactions. But for a variety of reasons, after Kochi's seminal work in the 1970s and early 1980s, this field remained dormant for about 20 years as the explosion of palladium catalyzed cross-coupling reactions dominated the field of cross-coupling chemistry. And it wasn't until the turn of the millennia when Alois Fürstner started to re-explore these kinds of reactions and reintroduce them, I think, to a generation of chemists that were perhaps not aware of the Kochi work that was being done in the 1970s. And I really like this Fürstner work and this example because it really shows one of the benefits of using iron-based catalysts for cross-coupling reactions. These kinds of reactions can be extremely fast. And in this particular instance, you can see we have this aryl halide and also an ester, and this Grignard reagent that's used in the cross-coupling reaction preferentially makes the carbon-carbon bond through the cross-coupling reaction mechanism rather than directly adding to an ester bond. And so this chemoselectivity is a consequence of the extremely facile cross-coupling reaction that is facilitated by the iron-based complex. And you'll notice also the temperature and the time for these reactions is quite is low and also short. And so these reactions also, conditions also illustrate how fast these reactions can be. Okay, in addition to these kinds of cross-coupling reactions, the Nakamura group became involved in this research about a decade ago, where they reported these very interesting cross-coupling reactions that were reminiscent of nickel-based cross-coupling reactions, where we can use sp3 hybridized electrophiles to then carry out these kinds of sp2, sp3 catalyzed cross-coupling reactions using these phosphine-based iron complexes. Once again, pretty efficient reactions and in good yields for these kinds of transformations. So while all of these reactions were excellent examples about how iron-based complexes can be used for cross-coupling reactions, you'll notice that there's a common feature to all four of these reactions on this slide, and that's that the nucleophile in these reactions are all Grignards. And so these so-called Kumada-Karash type, Kumada-Curio type cross-coupling reactions were pervasive in iron catalyzed cross-coupling reactions. And the industry preferred Suzuki-Mayura cross-coupling reactions where you use boron-based nucleophiles were largely absent from the literature. In fact, if you looked in the literature before our report a few years ago, there are only 2 examples of iron catalyzed cross-coupling reactions that involve boron-based nucleophiles. And these reactions had some interesting additives that were added to these reactions. Magnesium bromides required for high yields in these reactions, and also alkylithiums are required to activate the boronic ester by the alkylithium adding to the boronic ester to make a borate, either in situ or before the cross-coupling reaction. So it is very important to note that the mechanisms for iron catalyzed cross-coupling reactions is still hotly debated, and there's lots of questions about the steps in these particular transformations. Many people debate what the oxidation state of the iron complex is, and certainly the most interesting step, the carbon-carbon bond forming step, is also the one that is less understood in the steps in these catalytic cycles. So here, the Nakamura group and also some excellent work by Mike Neidig has delineated some of the mechanistic features for these reactions. And what happens, according to the Neidig group, is that you start with this iron dihalide iron 2 compound that undergoes transmetallation with the borate species that's formed from the alkylithium reacting with the boronic ester. Then this iron phenyl halide undergoes halogen atom abstraction, which leads to an iron 3 complex and the concomitant formation of a carbon-centered radical. Then, as I said, the carbon-carbon bond forming step is not well understood. There's many proposals. One possibility is a carbon radical rebound to make the carbon-carbon bond. Another is that the carbon radical recombines with the iron center to oxidize it to a higher oxidation state, which then undergoes reductive elimination to lead to an iron 2 complex. The other major camp for these mechanisms for these kinds of reactions is, instead of this iron 2, iron 3 mechanism, is a mechanism involving iron 1, so a low valent iron complex, that proceeds through 3 oxidation states, with the maximum oxidation state being iron 3. And the first steps are very similar. So you have a transmetallation event followed by halogen atom abstraction. The big difference here is that transmetallation events proposed to occur from the iron 1 intermediate, as well as the halogen atom abstraction, rather than from the iron 2 as proposed by Nakamura and Neidig. And so this mechanism has been popularized by Robin Bedford, where they use a very similar types of phosphine iron compounds. So these are the ligands that are used in these kinds of reactions. As you can see, they're not that much different from each other, but the two groups propose very different reaction mechanisms, especially with respect to the oxidation state of the metal complex. After the carbon halogen atom abstraction, once again, the carbon recombination here, the Bedford group proposes a radical recombination to make an iron 3 compound that then leads to reductive elimination. Okay, so this is kind of where we entered the research, and we were curious why it's so necessary, considering that iron cross-coupling reactions are so efficient in Kumata type cross-coupling reactions. Why is it necessary to soup up the boronic ester so much to make these borate transmetallating agents, and also magnesium additives are required for both the Bedford and the Nakamura catalytic reactions? So my group has been very as fond of this chemistry, and we are also very much aware of the excellent mechanistic work that had been emerging from a variety of groups over the last 3 or 4 decades on the palladium catalyzed cross-coupling reaction. And there's growing evidence that the palladium catalyzed cross-coupling reaction and Suzuki-Miura cross-coupling reactions involves these palladium hydroxide intermediates. And so the debate is always, what's the role of the base? And in this mechanism, after the oxidative addition event occurs, sodium hydroxide then combines with the palladium aryl halide to make the palladium hydroxide. And this palladium hydroxide then is sufficiently nucleophilic to then undergo the four-center transition state required in transmetallation, which leads to this penultimate palladium, or the ultimate palladium-2 diaryl compound that undergoes a reductive elimination. And so my group is very interested in synthesizing metal alkoxides and iron alkoxides for a completely different project in polymerization catalysis. And we hypothesized that these kinds of similar kinds of intermediates, iron alkoxides and iron hydroxides, are prone to aggregation. And these aggregation events, we hypothesized, would take the iron alkoxides out of the catalytic cycle, and therefore make it impossible for the transmetallation event to occur. So that was our working mechanistic hypothesis. But there was another competing possibility, and that is that when you move from palladium to iron, you significantly change the electronegativity of those two elements. And iron is an earlier transition metal than is palladium, and by doing so, iron becomes less electronegative than boron. And so traditionally in cross-coupling reaction, transmetallation rates are dictated often by the Schlenk equilibrium for transmetallation from the organometalloid to the transition metal species. And those transmetallation Schlenk equilibrium are often dictated by the relative electronegativity of the two elements. And so what my talented graduate student did was to look at transmetallation rates, transmetallation kinetic thermodynamics through calculations. And so he modeled these DPPE complexes that were popularized by the Bedford group to look at how thermodynamically disfavorable or favorable a transmetallation would be from an iron halide, an iron bis alkoxide, and then an iron alkoxide aryl complex. And as you can see from the iron halides, this kind of transformation is very much uphill. 80 kcals per mole for two transmetallation events. That is not going to happen for a reaction at room temperature. If, however, you convert those chlorides to phenoxides, or even better, electron-rich methoxides, then those transmetallation events become thermodynamically uphill, but not so significantly uphill to preclude a reaction that is occurring at room temperature. So 6 kcals per mole approximately per transmetallation event. And so we were encouraged by this fact, because overall the cross-coupling reaction is thermodynamically downhill, and we thought that this kind of barrier would not be impossible to overcome unless the kinetic barrier for these kinds of processes was particularly high. Okay, so what my graduate student did was to try to synthesize some of these discrete iron alkoxide complexes. And what we started with was the most obvious thing to try, which is this bisphosphine iron dihalides, and expose them to sodium alkoxides. And when Michael did this, he got this green insoluble material that we have not been able to characterize fully. It's literally a rock. It doesn't do anything. We can't even characterize it by Dosy spectroscopy. What we hypothesize it is this higher ordered iron aggregate species. And it's completely inactive for many things, especially important is if you expose it to the alkyl halide and boronic ester, you don't get any cross-coupled products. We were not surprised by this reaction, because in our polymerization catalysis work, we do not synthesize iron alkoxides using this salt metathesis route. And what we do normally is start with an organometallic reagent and then protonate the iron carbon bond using alcohols. And this we have found to be a much more general route to mononuclear iron alkoxide complexes. However, with these phosphine ligands, we found that when we did this kind of reaction, we did not see any, we still saw the same green insoluble material that was inactive for cross-coupling reactions. So what my student then did was to try to go back to the drawing board and think about what kind of functional groups could we use instead of the iron alkoxide that would be less prone to iron aggregation. And what my graduate student came up with was using iron amides instead of iron alkoxides. And here is the same computational method that we use to evaluate transmetallation with iron alkoxides. And what we were pleased to see is if you look at transmetallation from an iron amide instead of an iron alkoxide, this is no longer thermodynamically uphill, it's now downhill for that transmetallation reaction. Moreover, my student was able to find the transition state for this first transmetallation event and show that that barrier is very low, 14 kcals per mole, which means that this reaction should be feasible at room temperature. And so then Michael went back into the lab and tried to do these kinds of reactions instead of with alkoxide bases, with amide bases. And what he discovered was, instead of forming this green insoluble material, what he saw was a pale yellow solution that was homogeneous, and by NMR spectroscopy was one compound. This compound that we propose is this iron bis amide species is extremely reactive and difficult to isolate. So we don't have any further characterization data on that compound other than the NMR spectrum. But what is exciting was that if you take the boronic ester and the alkyl halide, you then start to see cross-coupled products. This stoichiometric reaction was extremely sensitive to the identity of the lithium amide base. So you can see here that dimethyl amide gave no yield of the cross-coupled product. The diethyl amide gave us about 40% yield of the cross-coupled product, and the methyl ethyl amide gave us almost 90% yield of the cross-coupled product. So this reaction is very sensitive to the steric environment about the base, which I will return to in a little bit. So we then proceeded to evaluate a wide variety of ligands. And so this is what is always done in this field, is people just kind of survey whatever ligands they have in their box and what's available to them. And that's exactly what we did. And what we found was a very useful class of ligands were these bisoxazoline ligands. And an important feature of these bisoxazoline ligands was this cyanide group that's installed in the back of the ligand. That's a very important aspect of this complex in order for it to give high yields of the cross-coupled reaction. So remember, these are stoichiometric reactions. In catalytic reactions, if we use this phosphine complex, we got about 30% yield of the reaction, which is about what we saw with just iron dichloride. So you do get some cross-coupled products from just adding iron dichloride with no ligand at all in these reactions. But here you can see a boost in turnover number by adding the ligand, this bisoxazoline ligand, to the reaction, cyanobisoxazoline ligand, and now we're getting 80% yield of the cross-coupled product using catalytic quantities of the iron-based complexes. Okay, so we then, of course, started to survey the scope of these reactions, and I'm kind of an anti-table person, so I'm exploring different ways of representing yield and things like this. So I haven't seen anyone use this yet, so I'm going to explain it to you guys very shortly. These targets kind of represent 100% yield. The solid portion of the yield is the actual yield that we obtain, the isolated yield in these reactions. And the dotted line indicates the yields based on recovered starting materials. And so by just looking at these targets, you can kind of get a sense for the reactivity of these kinds of compounds. And as you can see, the reaction proceeds very well with alkyl bromides as opposed to alkyl chlorides and iodides when you use unactivated alkyl halides. Moreover, instead, if you're using activated alkyl halides like benzylic halides, the alkyl chlorides actually are superior to the alkyl bromides. We didn't even try the alkyl iodides for fear of no reaction at all. And the reactions with alkyl chlorides are working so great. You'll notice in the reactions, especially here with the alkyl chlorides, what happens is we get good low yields, but very good yields based on recovered starting materials. So still, the major problem with these kinds of reactions is the catalyst dying in these reactions. And we think that that's because the ligand is still falling off in these reactions and iron aggregates are being formed. The reaction also can tolerate a variety of boronic esters. This took a little bit more reaction engineering, but at elevated temperatures or using more catalysts, we can also get good cross-coupling yields in those reactions. Okay. We then were working this was part of our work with a Pharmaceutical Roundtable, and one of the great things about working in the Pharmaceutical Roundtable is that we are paired with partners in industry. And being in Boston, there's many options for that partnership. And we were paired with Amgen for our Pharmaceutical Roundtable project. And as we were talking about this chemistry, one of our collaborators said, well, you know, cinecalcet may be a really good application for your cross-coupling chemistry. And this is a very simple molecule that you can make in short order and with many different methods. But all the methods that we could see in the Amgen patents for making cinecalcet or Sensipar involve some kind of cross-coupling reaction, palladium catalyzed cross-coupling reaction, or an olefin metathesis to make that carbon-carbon double bond. And then there was an iron catalyzed cross-coupling reaction that appeared in the literature, but it was an sp2 cross-coupling reaction. And then this hydrogenation reaction always involved a noble metal-based complex for the hydrogenation. And so we thought we could get rid of the hydrogenation reaction and also not use any of the metals for the cross-coupling or cross-metathesis reactions by using our chemistry. And taking advantage of the reactivity of the alkyl bromides over the alkyl chlorides, we can then do this reaction without much optimization. So I'm sure that we can get yields higher than this. I was pretty happy with just kind of this at the first shot that we can get 55% yield of this cross-coupled product, which then easily can couple with this amine, which is amazingly actually pretty cheap and commercially available to make cinecalcet in 2 steps instead of 3 like most steps, and not involving any of the noble metals that are typically used for the synthesis of cinecalcet. Okay, we have more recently been exploring an anti-selective cross-coupling reactions, and this is some unpublished work that hopefully will be published very soon. And here we found that using the chiral and enantiomerically enriched bisoxazoline compounds and benzylic halides, these reactions give us pretty good yields and decent stereoselectivities in cross-coupling reactions, especially for these sterically encumbered substrates, which give very high selectivities for the cross-coupling reactions. And this is particularly important because these kinds of substrates, actually, if you look at many drug molecules, these 1,1-diaryl alkanes often have orthosubstitution. And many of the known methods for making these kinds of stereogenic centers do not proceed in high selectivities, especially if it's a cross-coupling methodology. And so I think this is because the reactivity of the iron-based complexes is very high, and as such, you can access unreactive substrates. And so here's another scope with these kinds of reactions. And so some of these molecules, like this one, I can't imagine how you would make that with a hydrogenation route, enantiomerically enriched. And so we don't get awesome ERs like we get the sterically encumbered substrates, but still I think pretty respectable ERs considering the difficulty in that transformation. Okay, so I wanted to mention some limitations to our cross-coupling reactions. Academics like to talk about all the great things, but I think it's also important to mention some of the limitations. And one of them was the solvent, okay? And we were, of course, very much aware of this and somewhat embarrassed about this because we understand the importance of solvent in green chemistry. And for us, these reactions just worked really great in benzene. And some of the more useful solvents that are greener, like 2-methyl-THF and methyl-T-butyl ethers, gave us very low yields in the cross-coupling reactions. We were able to get pretty decent yields using anisole, which was some consolation, but this is not exactly a favorite solvent for students because it's difficult to remove, and they're sitting all day by the rotovap, and so they really didn't like that. My group loves it if we can use 2-methyl-THF because it kind of gives the best of both worlds. But this catalyst didn't allow us to use this, and so that was one limitation. Another limitation of this chemistry was that heteroaromatics weren't tolerated very, very much. And so you can see in these reactions, this thiophene and also this Inbach-protected indole gave us none of the desired cross-coupling reactions. And in fact, many iron catalyzed cross-coupling reactions do not have good tolerance to these kinds of heteroaromatic substrates, and that's a problem for pharmaceuticals because these are in a lot of pharmaceutical agents. And so here's a literature survey that my students did on the different kinds of cross-coupling types. So obviously sp2-sp2 cross-coupling dominates. sp2, sp3 are the second most common, and sp3, sp3 are the least common. If you look at the sp2, sp2 cross-coupling reactions in the literature, reported in the literature, by far, most of them involve aryl nucleophiles and aryl electrophiles. It's much more rare to find examples in the literature where cross-coupling involved hetero-aryl nucleophiles and hetero-aryl electrophiles. And that's kind of reflected what we saw in our Orglet paper. So the other problem with this reaction is if you use very sterically encumbered cross-coupling reagents like this tertiary alkyl halide, you can get some yields in the cross-coupling reaction, which we were very excited about at the time, because making these quaternary centers using cross-coupling reactions is very, very uncommon. As you can see here, this is for alkyl-aryl cross-coupling reactions, and these are the different types of electrophiles and nucleophiles that are used in those cross-coupling reactions. And as you can see, the reports where tertiary electrophiles or nucleophiles are using those in cross-coupling reactions is extremely rare. And so what we wanted to do was, instead of taking this kind of empirical approach, we wanted to try to start using logical ligand design principles to improve our catalytic reaction. And this has not historically been done in iron catalyzed cross-coupling reactions, certainly not to the extent that has been done in palladium catalyzed cross-coupling reactions. And here's our working mechanistic hypothesis for this reaction, where we have an iron halide that undergoes salt metathesis with a base to make the iron amide. And then this key transmetallation event then leads to an iron aryl complex, which then can undergo halogen atom abstraction to form this iron 3 compound that then somehow makes the carbon-carbon bond. Again, we're not sure how that happens, to then form a cross-coupled product and regenerate the iron 2 halide. So you'll notice one of the features of this ligand is that it's anionic, and that's a big difference in the phosphine ligands that were used previously, and we think that that's very important to prevent iron aggregation. So the phosphine, the neutral phosphine ligands have very weak bonds with iron. It's a much earlier transition metal than palladium, so a better match from a hard, soft Lewis acid base standpoint is anionic ligands for iron, and so we think that's very important. So in our ligand design, we wanted to have a bidentate ligand that would hold on to the metal center. We also wanted an electron-rich ligand that had steric bulk more central to the metal center. So these bisoxazoline ligands have steric bulk, but they're kind of far away from the metal center. So if you draw a space-filling model, you'll see that that is true. And finally, we wanted an anionic donor that would push electron density onto the iron center, because what that does is it makes this iron amide very nucleophilic, and that will allow it to participate in the transmetallation reaction. And so we surveyed the literature, and we were very familiar with these beta diketaminate ligands, or I like to call them NAC-NAC because it's a more fun word to say. It also sounds like AFLAC, and I always wanted to say it. So I really wanted to use this chemistry, partly because I just want to say NAC-NAC a lot. Anyway, these NAC-NAC compounds, we are very big fans of Pat Holland's group, and these iron complexes with NAC-NAC ligands on them are very reactive compounds and can do some very impressive reactions. These iron compounds are known to reduce nitrogen gas to ammonia. And so we thought, look, if these compounds can reduce nitrogen to ammonia, a boronic ester, it has no chance with these kinds of compounds. And so my student did some beautiful organometallic chemistry that I'm going to whiz through here because I'm running a little bit short on time. But what he did was able to synthesize these discrete iron alkyl complexes that he could then transform to iron amides. He now has crystal structures of these iron amides that are much more amenable to isolation and characterization than those with the phosphine on them. We can also show that even though these are dimeric in the solid state, using this really cool technique called Dosy Spectroscopy, which my graduate student pioneered for paramagnetic compounds. These are all paramagnetic compounds. You can do NMR spectroscopy on paramagnetic compounds. And here you can see in the Dosy spectrum one peak for the iron complex, which it has a molecular weight. You can use that diffusion coefficient and correlate that to the molecular weight of compounds, and it has a molecular weight that's more consistent with a mononuclear species. So that we believe in solution that these compounds exist as an equilibrium between a dimer and a mononuclear species. And the dimer is what crystallizes out in the solid state. Okay, he could then take that iron amide and expose it to the phenyl B-pen compound and form an iron phenyl compound, which is also isolated and characterized by NMR spectroscopy, and here, X-ray crystallography. And this is a really cool bridging iron phenyl complex, which we also believe to exist as an equilibrium between dimeric and mononuclear species in solution, although we have not done the Dosy experiment on these compounds. What's exciting is if you take this iron phenyl compound and expose it to the alkyl halide, you get very clean production of the desired cross-coupling product and also forming the iron halogen atom, which is how we started this whole process. And so then Michael then looked at a whole bunch of NAC-NAC ligands. So these are very easy ligands to synthesize. Unlike the bisoxazoline ligands that take my students a lot longer to make, the NAC-NAC ligands library, they synthesize, I think, 20 of them in about a month between 2 graduate students. And so, you know, at your companies with like the team army of people that you have working, I'm sure that you can generate a huge library of these ligands. It's important that you make the complex first. We do not get good reactions if you just throw in the ligand with an iron precatalyst. That doesn't work very well, and I can talk about that in the Q&A if there's time. And what's exciting is that as you change the steric environment about the metal complex, you then tune the reactivity of the catalyst. And so if you have too sterically encumbered of a NAC-NAC ligand, you don't really see any reactivity at all. But for very sterically unencumbered aryls, now the steric environment is very close to the metal center, not a carbon adjacent to it, but on the nitrogen atom. You can see that we're getting very good reactivity such that it's 8 times as active as these disopropyl substituents, and now the half-life is about an hour. These reactions with the box ligands, initially they're very fast, but then they take forever to get to high completion. And so after 4 hours, you see significantly slowing. And so in order to get to these high yields, with these complexes, you need to carry out the reactions for at least 24 hours. But with the NAC-NAC complexes, they are now done in a few hours. Okay, so we were able to address some of the limitations that existed with cyanobisoxazoline ligands by changing them to the NAC-NAC compounds. And so here's a reminder of our solvent screen, right, that we obtained with the bisoxazoline ligands, but through catalyst design, now with a much better ligand for iron, you can then start to see reactivity that now, here's that 2-methyl-THF that I was talking about. My students are happy because they can use a green solvent and they don't have to spend all day waiting for the anisole to be removed from the reaction. And so we were very excited to see that we could address that limitation. Moreover, we were able to address the limitations with respect to the heteroaromatic nucleophiles as well. So here we got, I would say, disappointing results. But when you use the NAC-NAC compounds with the right substituents, you can then start to see very good yields for the cross-coupling reactions for those same kinds of substrates. And in fact, with help from Amgen, we were able to look at a variety of heteroaromatic nucleophiles, and we often saw good results. Some of them are not so good, especially pyridines. We found not to be very hit or miss. So some were great, we got very high yields in cross-coupling reactions, and others were not so great. And most of the ones that gave us great yields were those that had ortho substituents on them. So these often gave better yields than those that did not. But we certainly have examples that I don't show on the slide where we don't have that trait, and where there are ortho substituted pyridines and they don't give us high yields, as well as the alternative where there are not ortho substituted pyridines and they give us high yields. So it's still unclear what the deal is with the pyridines, but we can use pyridines, we can use piperidines, we can use indoles and thiophene, indoles, thiophene, and also what are those? Furans. Sorry. Okay, so finally, I wanted to share with you our reactions with tertiary alkyl halides, which there's our original yields, and then with the tertiary substrates, we get much higher yields in these reactions. And what I wanted to point out in the last minute here is the advantage of iron catalyzed cross-coupling reactions. So there are many people asking, well, what's the deal? You can just use nickel for all these reactions. Why not just use nickel? I said, well, in addition to the toxicity and availability concerns, many times nickel catalyzed cross-coupling reactions, here's a cross-electrophile cross-coupling reaction, they produce the desired cross-coupling reactions with these sterically encumbered substrates, but they also often produce isomeric products, and these are sometimes difficult to separate from each other. And we don't see that with the iron catalyzed cross-coupling reactions. We only see the desired cross-coupling reaction or no reaction at all. And the other thing that we've noticed is that there seems to be a slightly larger substrate scope with respect to the boronic ester. So here's the state of the art, as far as I'm aware, of nickel catalyzed cross-coupling reactions with tertiary alkyl halides using Suzuki type reactions. And yet, as you can see, you have to use a borane, and also the substrate scope with respect to the boronic ester is very limited. In contrast, here we can use a boronic ester rather than the much more pyrophoric boranes, and we also get a much broader substrate scope with respect to the boronic ester. Greg definitely does better with respect to the alkyl halide than we do. So that substrate scope is much better than ours, I will admit. But I do think the reactivity of this has to do, the difference has to do with the higher reactivity of the iron-based complexes that I highlighted with the Firstman group. Okay, with that, I'd like to thank all the researchers who did this work. Dr. Michael Crockett, he just defended his thesis a few weeks ago. Here he is right here in the front. I'm very proud of Michael. He took this project from basically nothing and made it something. Chet Tyrrell, along with Connor and Nancy, worked on the anti-selective cross-coupling reactions. Alex is the champion for all the heteroaromatic cross-coupling reactions. And Marcin is a first-year graduate student who's doing some really cool chemistry that I was hoping to share with you today. But because our lab was shut down for 3 months, we really didn't have an opportunity to do some of the great chemistry. And it's not quite ready for prime time with Marcin, as well as some things that Chet are working on. I couldn't do that. We could not have done this work without our partners at Amgen and also the ACS Green Chemistry Institute. This was a great partnership, and I really think that we benefited tremendously from understanding the issues that the pharmaceutical industry has with cross-coupling reactions and how we can make our reactions better for these reactions. More recently, this work is being supported also through the ACS and the ACS Petroleum Research Fund. We are actively pursuing federal funding, but we would be happy to collaborate with any industrial collaborators. Sadly, our collaboration with Amgen ended last year with all the, I'm sure everyone's aware of all the layoffs that happened at Amgen, and one of the casualties for that was the cession of this collaboration. So with that, I'd like to, if there's any time, I know I'm pretty late probably, but I'd be happy to take any questions if we have time. Thank you for your attention.
Thank you, Jeff. Excellent presentation. Certainly a great example of the evolution from one catalytic system where you actually address how to make the ligand, but especially how to address the on-green solvent usage at the beginning. So thank you again. Maybe you have time for one question. You mentioned about this type of NAC-NAC ligands to be easier to make. What about stability? How do you look at the stability of the ligand for the long term?
We haven't explored the long-term stability of the ligands. I know that we've made them now. I'm pretty sure that they've been on the bench for months now without any problems. I guess hydrolysis is what you would be concerned with, but certainly they're much more air stable than a phosphine ligand. They don't undergo oxidation reactions, and so we haven't observed any stability problems with the NAC-NAC ligands. But to be fair, we haven't specifically studied that.
Okay, well, thank you. Thank you again, and we will have more time at the end of the session to ask other questions. So now let's move to the final speaker, who's going to bring all of us home. So our next speaker is Dr. Frank Gupton. Frank, a good friend and an old friend from the Pharma Roundtable, and he is professor of the Virginia Commonwealth University, and he holds joint appointment in the Department of Chemistry and Engineering for the Life Cycle Engineering Department. He is the Floyd Gottwald Chair of Pharmaceutical Engineering and also served as the department chair of the Chemical and Life Science Engineering Department. Every time I read all this, Frank, I wonder how much you have time for so many other stuff outside of all this. But in his 30 years industry career, he has centered on the development and commercialization of chemical processes and pharmaceuticals application. Frank's group focus on the development of continuous processing technology to facilitate the discovery, development, and commercialization of drugs products. Prior to joining the faculty at the Virginia Commonwealth University, Frank served as Executive Director of North American Process Development for Boehringer Ingelheim and led the commercialization of widely prescribed HIV drug, nevirapine. He received his bachelor's degree in science and chemistry from the University of Richmond and graduated in organic chemistry for Georgia Tech and Virginia Commonwealth University. Currently, his research efforts has focused on the streamlining pharmaceutical processes in the area of active pharmaceutical ingredient by employing principles of process intensification, which include the use of innovative chemistry, novel continuous manufacturing platform, and the new, more efficient catalysis for pharmaceutical application. In 2018, Frank received the award for the affordable chemistry, and in the same year, he received the Presidential Award for Green Chemistry. In 2019, he received the Peter J. Wong Award, as we mentioned it, and among other many awards that he had received. So back-to-back award, 3 of them very important in the area of green chemistry. All of these associated with the great effort that Frank have done on developing better processes for delivering and giving access to drugs to the community. Today, Frank will be talking about the pharmaceutical processing in the 21st century. With that, Frank, please.
Thank you, Juan. I'm hoping you guys can see my screen now. Is that visible to you all?
Yeah, it looks
Great.
good, Frank. Yeah.
Okay, thank you. Juan, thanks very much for the introduction and also for the opportunity to speak with you today with this austere group of lectures. So I thought I'd start off by kind of framing the challenge that we're faced with right now with pharmaceutical manufacturing in the United States. And in that vein, I wanted to talk about events versus processes. And so one of the things that I think about as far as one of the major advances that occurred in manufacturing is Henry Ford's assembly line approach to the manufacture of automobiles. and this has been rapidly adapted by most of the products that make up the gross domestic product in the United States. However, pharmaceutical manufacturing hasn't really embraced this to that extent. And although it represents approximately the pharmaceutical sector and the related businesses around it represent approximately 17% of the gross national product, most of the work that is being done is event-based processing. And when I say that, I mean there's a beginning and an end to the manufacture of the process, as opposed to a continuum that you see in the assembly line approach. Now, pharma efforts in this area have been successful to a limited extent. And one of the things I'll show is GSK's effort to produce a drug that they have that's one of their main products. And this is a unit that does 3 chemical steps in a single footprint and is totally continuous to produce the drug API. So, with that, you start thinking about the fact that the chemistry does drive the complexity of the process. It affects all the unit operations, obviously the environmental impact, the capital cost, the operational cost, and ultimately the return on investment for these types of operations. So Juan mentioned earlier that our group is specifically focused on process intensification, and our group kind of defines that as any process change that leads to substantially smaller, cleaner, safer, and more energy efficient technology. And so obviously increasing safety and productivity, capacity and flexibility is a key, as well as reducing energy waste and the complexity of the processes that you're running. So when you look at what our research group is doing, these focus on not only the chemistry, but the equipment, and looking at the raw materials, how we can use very simple building blocks to produce these products. And at the same time, we're minimizing waste and whenever possible, using catalysis to promote these transformations. Now, there's a lot there, and when you start thinking about it, this is really at the interface between chemistry and chemical engineering. And I'm very fortunate to have dual appointments between chemistry and chemical engineering. I'm an organic chemist, but my research groups have typically been a 50-50 mix of chemists and chemical engineers. And having a joint appointment between chemistry and chemical engineering and serving as the department chair in chemical engineering has given me the opportunity to build a research group that is a very eclectic organization that's a mixture of chemists and chemical engineers that work together to solve these fundamental problems. So with that, I'd like to take a second and digress and talk a little bit about the 3 buckets of work that we're involved in right now. On the left-hand side, this is a slide that I typically go to explain to people the diverse nature of the research that we're working on. We've been working with the Bill and Melinda Gates Foundation now for about 6 years and looking at how we can increase access to global health drugs by improving the chemistry that's used to make the active ingredients. And we've initially focused on HIV drugs, and we've completed basically reinventing the chemistry for all the first-line therapies for HIV drugs. The second area that we were working on is a Center for Rational Catalyst Synthesis that's funded by the National Science Foundation. And it's something that's a bit foreign to a lot of the work that's being done in pharma, in that we're looking at figuring out how we can, instead of using homogeneous catalysis, use heterogeneous catalysis to be able to do some of the typical transformations that are occurring right now in the pharmaceutical industry. And lastly, we've been working for the last few years on a project called Pharmacy on Demand in conjunction with Tim Jamison's group and Klaus Jensen's group at MIT. It's a unit that's about the size of a large refrigerator. For those of you who don't know about this project, you put the chemicals in the front end, it synthesizes the active ingredient, formulates it, pills come out the back. And we've developed a continuous process to make Cipro, and we're in the process of receiving FDA approval and qualification for that unit. So with that, you start thinking about each one of those different areas. And the common theme in each one of them is process intensification and how these things can be applied to continuous processing. So again, just to kind of ground you on each of you on the difference between batch and continuous, the analogy I like to make is making spaghetti. So if you look at the process to make spaghetti sauce, it's a batch process. Typically, you're using small volume and higher value added products or starting materials. The operating costs per kilo are pretty significant because of the lower throughput, but it does have greater flexibility. You can produce a lot of different sauces in that one pot. Therefore, it's a multi-purpose piece of equipment. It can have a much higher capital cost, and the other thing is that's really important is the greater variability in product quality, and I'll talk about that in a second. And you contrast that against making pasta. It's very high volume and it's a low value added product, lower operating costs, limited flexibility because you can only make pasta in it. It's usually dedicated to a limited number of products, has lower capital costs because you don't need a whole lot of pots, you only need one unit. But most importantly, you get very consistent product out of it. And when you think about it, each batch of spaghetti sauce tastes a little bit different, but all the pasta tastes the same. And that's okay when you're making spaghetti, but not when you're making pharmaceutical products. And this is one of the reasons why the FDA has embraced this approach, is that the ability to develop a much more robust and reproducible process using continuous platforms. So with that, I'll take a second and talk a little bit about how this interfaces with our work with the Gates Foundation. So access to global healthcare is a real problem on the African continent, and particularly with regards to HIV drugs, malaria drugs, and tuberculosis drugs. But then there's another element to this related to environmental sustainability, and this is kind of the focal point for Medicines for All, is how we link those two together. And I'll take a few seconds and explain to you why this issue about environmental impact is so important relative to global access. So as many of you may know, there are more than 300 drugs currently in short supply in the US today. This was long before the COVID-19 pandemic. This is something that's been a sustainable problem in the United States for some time. Approximately 80% of all of our medications consumed in the US are produced in India and China, and I think that there's been an increased awareness of that in the last 6 months with regards to the supply chain discussions that are occurring in the federal government right now. And then lastly, this was mentioned earlier by Mike Kopak, that commercial API process produces between 50 and 100 kilograms of waste per kilogram of product. Roger Sheldon gave me this number, but I believe Mike's number is much more accurate. It's more like about somewhere between 100 and 200 kilograms of waste. And we've seen several processes when I was in industry that were well above that those numbers. So how are these three factoids related? So these are all related to our supply chain vulnerability, and the important developments that have occurred recently in China, not about disregarding COVID-19, are related to pollution control. So where does the waste come from in an active pharmaceutical ingredient process? Well, this is a schematic of a typical API plant where you've got a 4-story building, and on the 4th floor, all the chemistry takes place. Then it's fed down into a crystallizer, and then after the product is crystallized, it's separated in a centrifuge and then dried in typically a rotary tumble dryer. So if you look at the raw material charges, approximately 50 to 75% of that mass in that reactor is associated with solvent. And it comes out as solvent and byproducts in the centrifuge, as well as in the drying operation. And this is the major source of process waste in these pharma processes. So if you had 100% conversion in the 4th floor reactor, you'd still be generating at least an equivalent amount of waste or more of solvent waste that you'd have to deal with if you had a clean reaction. But the problem is usually we don't have clean reactions and the yields are a little bit lower. So the question we asked ourselves as a group was, why do we isolate intermediates? And the main reason that we do it is to purge impurities from the process so they're not carried forward into the reaction mixture. So this is a really important element of what we've been looking at. So if you look at the major objectives of the Medicines for All Institute is to expand global access to life-saving medications by finding new ways to make them more efficiently, by using new synthetic methods, novel manufacturing platforms, addressing the environmental impact, and looking at commercial implementation partners that can actually adopt these processes very quickly, and then having a more fundamental understanding of the drug product life cycles. And I'll go back and emphasize this issue about environmental impact, because what's happened is, in its relation to commercial implementation, is that over the last few years, the Chinese government, who's been making most of these drugs now, have implemented a thing called the Blue Sky Law. And over the last 3 years, they've shut down numerous pharmaceutical operations because of the waste that they've been generating. And that's resulted in higher costs of the active ingredients and more drug shortages in the marketplace. So this is one of my go-to slides, and it's kind of the boundary conditions for working in my laboratory. My graduate students really do understand that this is an important element of what we're doing. So the first thing that we look at is to ensure that we have high-yielding reactions, going back to this issue about isolating intermediates and the impurities that are generated. So typically, we will not accept a reaction that's under a 90% conversion. We use catalysis and we use convergent strategies to be able to put the molecules together more efficiently. We use compatible reaction conditions in these reactions once we've established that we can achieve high yields. And what that allows us to do is to look at common reagents that we can use and common solvent systems so we can telescope these steps into a minimal number of unit operations. The other thing that we do is something that's counterintuitive in the pharmaceutical industry, and that is typically the pharma processes want to acquire more advanced starting materials so that they can produce the active ingredient in a minimal number of steps. What we do is we go back in the process to very simple commodity-based starting materials and building blocks that are less expensive and therefore achieve our objectives with regards to increasing access to these drugs. And as I mentioned earlier, using limited unit operations, limiting the number of unit operations by telescoping these steps is kind of the final step in the process. But when you think about it, the high yielding reactions is really the most important activity that drives this effort, and limiting the number of unit operations is an outcome opportunity that we get out of this. And as I've said in the past in other talks, that this is really common sense, that that we should be doing these things. And I tell my students that all the time. And one of the quotes that I like to use is one from one of my graduate students. He says, "Common sense is not a gift; it's a punishment, because you have to deal with everyone that doesn't have it." And he's currently in the pharmaceutical industry, and he's doing great things. And I'm sure that he is passing this on to his colleagues. So with that, I want to talk a little bit about global access to pharmaceuticals to try and frame the magnitude of the problem that we're faced with in that space. Currently, there's more than 35 million infected HIV patients, and most of them are in Africa, about 25 million of them. We typically get over 2 million new infections every year, and about 1.5 million deaths. There were also, in 2016, there were 216 million malaria cases reported on the African continent. And again, most of those were in Africa. And then the other one, it was something that we don't really think about too much in the US, is tuberculosis. And there was more than 10 million tuberculosis cases reported in and most of those were in Africa. And I think the other thing that the Gates Foundation pointed out to me when we were working with them is that this is where all the population growth is going to occur over the next 10 years. That they're expecting that on the African continent, the population will double over that period. And this is basically the only place in the globe that's going to have a significant increase in population. And it's in the midst of all of this massive amount of infection that's occurring on the continent. So we feel this is an unmet need in our time that we need to address. So very simply put, I thought I'd put this slide in to show you how we work with the Gates Foundation. We've been developing, once the Gates Foundation selects a target for us to work on, we spend a significant amount of time using the principles of process intensification to develop novel chemistry that we then transfer to manufacturers. And then the Gates Foundation monitors the cost of those drugs in the marketplace and the impact that we've had on these new processes. And I will say that unequivocally, one of the major reasons for the rapid uptake of these new processes is the reduction in waste generation in the processes due to the pressure that's being created in China over reducing waste generation in overall in their country. So this is basically a list of all the drugs that over the last 6 years we've developed processes for. Most of them have been done both batch and continuous. I will mention that the Gates Foundation is interested in getting these processes out as quickly as possible, which means that typically we'll use batch manufacturing platforms to do that. But all of these processes that we've developed are basically agnostic with regards to the manufacturing platform and can be produced either batch-wise or continuous. So the two I wanted to just speak quickly about are ones that I've talked about in the past. One is nevirapine and the other is ciprofloxacin. Nevirapine is the first non-nucleosidic reverse transcriptase inhibitor that was used to treat HIV-infected patients. It's been a member of the combination drug therapy for since the mid-90s and is really an effective agent in the treatment of perinatal HIV. When we launched this drug in 1996, approximately 30% of the women of childbearing age on the African continent had HIV. So the probability of newborns getting that was extremely high, and being able to treat the newborns with this with nevirapine really was an effective way of dealing with that outcome. So if you look at the linear process to make nevirapine, it's basically a linear method for production of the material. It requires multiple solvents. The overall yield, when I say it's low for multiple steps, it's about 59%, which isn't bad. It's about 6 unit steps and 21 unit operations, and the market price has varied. When we started the process was between $135 and $225 a kilo. And one of the key features on this process was that in step 3, when we form this diazepine ring, we use sodium hydride and diglime as a solvent. And one of the things we recognized was that if we used any other solvent or base, that we got an entirely different product. If you look at this intermediate cyclor, there are basically 2 acidic protons on it. One is on the amide proton, and the other is on the cyclopropylamino group. And the one, it's necessary that we deprotonate the cyclopropylamino group to do the ring closure. But the fact that the amide proton is more acidic, if you use any other solvent, you end up with an oxazole rather than the diazepine ring system. So we knew that we were kind of restricted into what we could do upstream if we wanted to telescope the process further. And so we started to focus on ways of being able to put the cyclor together using these same conditions of sodium hydride and diglime. So the new process looks like this. So we basically take this starting material, 2-chloro, 3-amino, 4-picoline, And we reacted it with sodium hydride and diglime and deprotonated it. And then we reacted it with the ester of the nicotinic acid moiety. And that gave us the amide in very high yield, high conversion. And then all we needed to do was to add another equivalent of sodium hydride and diglime, and we made the crude product in a single pot. We were able to telescope the synthesis. We used a single solvent, and we took the yield from the high 50s to approximately 92%. And in doing so, we reduced the raw material cost to about $60 a kilo. So when you start visualizing what the process looks like, this was the first generation process. And I might point out that we generated multiple impurities in this and required an additional crystallization, or a recrystallization to be able to purge the impurities from this process. We then came up with a second generation process that looked like this, that avoided that second intermediate recrystallization due to increasing yields. But this is what the new process looks like. And you can see that we've dramatically reduced the number of unit operations. And looking at our process metrics, we were able to increase the yield to from the 50s to the 90% yield in the overall process. We also were able to significantly reduce the number of unit operations by about 2/3. And but equally important, we had a huge reduction in the amount of waste that was generated in the process. If you include the amount of water in the system, we were right at about 9 kilos of waste, at a PMI of 9 versus a PMI of about 56. So this is what we could, an example of what we could do with processes, existing processes that could actually be re-engineered and made to be more efficient. So Gates Foundation, using this model, transferred it to one of their 2 commercial manufacturers. And we immediately saw about a 20% reduction in the cost of the active ingredient. And one of the things we did do is the overall cost reduction was about 40%. But by sharing the cost savings with the manufacturer, it actually helped to strengthen the supply chain. So that was an additional benefit that we got out of that. So looking at the impact, we've gone through virtually all of these high volume HIV drugs and developed new processes for them. So on the left-hand side are the different current regimes for treating HIV. And when you add this, an average savings price of about 10% to each one of the active ingredients that are going into the product, it represents an annual savings of somewhere between $85 and $90 million, and the Gates Foundation has validated that. So they consider the investment that they've made in the Medicines for All Institute as something that's financially viable and gives them an adequate return on investment. So I'll take a second to turn to our work with Pharmacy on Demand. And I mentioned that earlier, but the drug target that we were working on was ciprofloxacin. It's a quinolone-based antibiotic that's an essential medicine identified by the World Health Organization. And the innovative drug company was Bayer on this. So if you look at the process to make Cipro, it very